Hello everyone. Today I want to discuss about the thalassemias. Firstly, I want to discuss this topic under two parts. First part consisting of classification of thalassemias. The next part consisting of talking about molecular basis of thalassemias and clinical diagnosis of beta thalassemia major. So this is about the structural representation of the hemoglobin, where heme and globin forms the main counterpart, main part. So hemoglobin is the main constituent of the red blood cell. Iron is the main constituent of heme, and it bonds to the globin, where it has the alpha and beta chains. Normal human hemoglobins are of these types, and the structural formulas are as this follows: where adult type of hemoglobin is of majorly this hemoglobin type A, and structural formula is alpha to beta 2, and it constitutes 97%. Whereas there is also other type of hemoglobin that is in adult life, that is hemoglobin A2, which is structural formula of alpha 2 delta 2. Then constitutes 1.5 to 3.2 to 1.5 to 3.2%. Then in fetal hemoglobin are of HbF, embryonic life weight as HB over 1 over 2 Portland. So this is about the graphical representation of types of hemoglobin in prenatal and birth condition and postnatal. Coming to the introduction of thalassemias, term thalassemia is derived from the Greek word thalassa, which means sea. This disease was described by the Thomas Cooley, and its ICD classification is D56. Definition of thalassemia: thalassemias are a group of inherited disorders of hemoglobin characterized by reduced or absent production of globin chains. So this is about the globin chains deficiency. So the classification of thalassemias based on type of globin chain which is deficiently synthesized, it is classified as alpha thalassemia, beta thalassemia, delta beta thalassemias, gamma, beta, delta thalassemias. Molecular basis of thalassemia, so its common types are alpha thalassemia, beta thalassemia, where alpha thalassemia occurs due to the gene deletion, whereas beta thalassemia occurs due to the point mutations. So alpha thalassemia, which occurs due to the inherited gene deletions, can result in conditions such as hydrops fetalis, can result in HBS disease, alpha thalassemia trait, alpha thalassemia carried states. Whereas beta thalassemia, which occurs due to the point mutations, can result in following conditions such as thalassemia major, thalassemia intermediate, thalassemia minor. Here, the important point to remember is on two chromosomes, that is from paternal and maternal chromosomes. Each chromosome has two alpha genes and each chromosome has two alpha genes. That is paternal chromosome has two alpha genes, paternal chromosome has two alpha genes. I'm talking about the alpha thalassemia. The alpha thalassemias are caused by the inherited deletions that result in reduced or absent synthesis of alpha globin chains, already mentioned earlier. So this can result in clinical symptoms such as silent carrier state of alpha thalassemia trait, HBS diseases, hydrops fetalis. Silent carrier state of alpha thalassemia can occur due to the deletion of a single alpha globin gene, can be in paternal or maternal chromosome. The individuals are completely asymptomatic but have slight microcytosis. Alpha thalassemia trait. It is caused by deletion of two alpha globin genes either from a single chromosome or one each on the two chromosomes. That is, two chromosomes, one from paternal, one from maternal. Each chromosome has two alpha globin genes. If two alpha globin genes are related on either paternal or maternal, can result in alpha thalassemia trait, or one each alpha globin gene related from the paternal maternal chromosome can result in alpha thalassemia trait. The clinical picture is same, but the inheritance is different for both these types. The clinical picture includes microcytosis, minimal or no anemia, and no abnormal physical signs in alpha thalassemia trait. Decreased MCV and MCH can be seen in alpha thalassemia trait. Definitive diagnosis requires globin chain synthesis studies and genetic analysis. Coming to the HBS diseases, it is caused by the deletion of the three alpha globin genes. Synthesis of alpha chains is markedly reduced and tetramers of beta globin called as HBH are formed. HBS has high affinity to low oxygen leading to tissue hypoxia. That is HBH when disease when occurs. This type of HBH hemoglobin has high affinity to oxygen, which leads to the tissue hypoxia. This is a pictorial representation of the tetramers of HBH, which is formed leading to the sequestration of the RBC in spleen, causing moderate to severe anemia. So this is the arrow mark representing the HBH inclusions which are seen. Coming to the heteros fetalis, it is the most severe form of alpha thalassemia where it is caused by the all the globin alpha globin gene deletions. In the fetus, excess gamma globin genes chains are formed with cells tetramers which have high affinity to oxygen and deliver little to the tissues. Sign of fetal distress usually becomes evident in the third trimester. There is lifelong dependence on blood transfusion for survival. However, hematopoietic stem cell transplantation can be curative, but is not guaranteed. Coming to the beta thalassemia, clinical classification of beta thalassemia based on severity of anemia, which in turn depends on the genetic defect, where it caused due to the point mutations of the beta globin genes, where beta null or beta positive, and the gene dosage. Individuals with two beta thalassemia alleles have a severe transfusion dependent anemia called as beta thalassemia major. Heterozygotes with one beta thalassemia and one normal gene have a mild asymptomatic anemia, and this condition is referred to as beta thalassemia minor or trait. Third heterozygous variant of moderate severity is beta thalassemia intermedia. So this is up to now. I discussed in the about the structure of the hemoglobin and how the alpha and beta thalassemias are classified. Next part we will discuss about the molecular basis of thalassemia and discuss about the clinical diagnosis of the or the lab diagnosis of the beta thalassemia major. Thank you very much.